Okay, so can you guys tell me a little bit about what your experiment is on?
Our experiment is on the effects of arthrobacter and the decomposition rate of polyethylene in microgravity.
Okay. So did you all encounter any problems when you all were doing this experiment?
There definitely was some problems just because we're coming from a high school, so trying to get a bacteria like arthrobacter, they don't usually send that to high school, so you need to be at a certain level to get that, like a university or college. So we were able to go to an institute, and then we got the bacteria there.
Is this experiment ready to fly, or has it flown before?
It has already flown.
How was that?
It went really well. It actually has flown twice. The first time, they didn't necessarily crack the cartwheels the way they were supposed to, so it had to refly again, and then we got our results back, and it went well.
Okay, well that sounds great. I wish you guys the best of luck.
Thank you very much.
Thank you.
So what exactly is your presentation about?
So basically what we did is we looked at the bacteria Pseudomonas aeruginosa and how microgravity actually changes its resistance to antibiotics in order to kind of determine when we already have humans in low Earth orbit constantly, and when we eventually colonize space, how will disease treatment change?
Has this been tested in space already? Has it been flown before?
Our experiment has flown, but we've had some confounding variables that allowed us to get another flight from NASA.
What were some of those confounding variables?
So basically what happened is the design was to expose the bacteria to a growth medium for a few days and then a growth arrestor so that we could stop bacterial growth before the colonies would die out. But what actually happened due to a mishap is that the bacteria was exposed to the growth medium and the growth arrestor simultaneously. So we predict that little growth actually occurred aboard the space station. So we're not sure if our results are 100% accurate because the bacteria wasn't actually grown in microgravity.
So what are the next steps for this project?
So we're gonna relaunch it in the fall, which we're really grateful and we're really lucky. And that also really gives us the opportunity to restructure our experiment a little bit.
Also, another thing we were planning to do is use more types of antibiotics. In our original experiment, this experiment, we only have 3 different antibiotics. We plan to test more.
Okay.
Thank you.
What's your experiment? What are you guys testing for?
Basically, we're testing an experiment based on ALS patients. And ALS, what happens is that the motor neurons break down because of a substance called glutamate. It builds up and it's not broken down properly. We're simulating that reaction and we're breaking down a protein with an enzyme, and we're seeing that if that reaction is more efficient in microgravity situations.
Okay, so if I have this right, you're testing for the efficiency of these enzymes in a microgravity state.
Correct.
And do you guys have a hypothesis? Do you think it will be more efficient, less efficient, or are you guys just...
More efficient, because it turns out that in microgravity, we're hoping that since in gravity, the enzymes can't move as well as they could in microgravity, because in microgravity, we're hoping that there'll be more collisions between the enzyme and what we call the substrate, which is what the enzyme's going to be breaking down. So we're hoping there'll be more collisions and more substrate will be broken down.
Yeah.
All right. So what experiences have you done on Earth, or have you done any to sort of lead you to this general experiment?
We actually, as far as our mission hasn't gone to space yet, we're actually flying, is on mission 3, which is going in November. So the main experiments that have been done on ground is kind of determining the amount of enzyme, which is a papain extract, and the fixative, which is kind of a substance that's going to terminate the reaction, which is called vitamin C. So we're the experiments are just seeing which amounts of papain and vitamin C should be used, and we decided on using 0.2 grams of papain and 0.2 grams of vitamin C, and then using those amounts, we kind of just did some ground trials, some trial runs with the experiment and with the FMEs to just get results.
And then we'll compare these results to the space results that we get, and then we'll see if it's more efficient.
Okay, very good. And do you foresee this having any benefit for astronauts or just people as a whole?
We're hoping for people as a whole, because what happens is that every second we're getting closer to commercial space flight, and there are many diseases that affect the enzymes in a person, and a certain kind of enzyme, and it doesn't work as properly. And what we're hoping is that one day we'll get to the point where commercial space flight is relatively cheap, maybe 20, 30 years down the road, and we can send up patients that suffer from these kind of diseases, and we can prolong their lives or hopefully even make it so that they could be cured.
To see if it ease their too.
Yes.
Yeah, or even on the other hand, we could see if we could maybe have technology enough to simulate a microgravity environment here on Earth. So it depends on how down the future. That's all in the future.
Yeah, it's something looking ahead to the future.
Yeah.
All right, very good. Thanks for your time.
Thank you very much.
The effectiveness of hydrogen peroxide and astragalus nitrogen growth in microgravity.
So what experiments did you conduct on Earth?
With the ground control, we had sort of two ground controls. One, we would just do the experiment that was going into space, except not in a test tube, but on Earth. And we had with hydrogen peroxide. The second one, we were just going to do without hydrogen peroxide and just see if water would dilute it. And we were comparing the differences between the hydrogen peroxide ones and the ones with just water.
All right, and what is the biggest problem y'all came across during this experiment?
When we got the results back, the biggest problem was that it got contaminated. When she touched this to the opening of the valve, the FME leaked, so the mold got transferred to the outside of the tube, and when it came in contact with the other tube, it contaminated it. So
Did this experiment fly already, or is it expecting to fly?
it flew on October 7th.
All right, well, I wish you guys the best of luck with your experiments in microgravity environments.
Okay, so can you guys tell me the purpose of your experiment?
We are trying to figure out if bones will absorb calcium in microgravity.
Okay, so how did you go about completing this type of experiment?
We are going to weigh, measure, and micro CT scan the control and experimental bones first, and then we're going to decalcify them in distilled water or vinegar, and then we're going to weigh, measure, and micro CT scan them again, and then we are going to soak the decalcified control and experimental bones in buffered saline calcium solution for the same amount of days. And then we're going to weigh, measure, and micro CT scan them again. And then we will compare the weights measurements and micro CT scans and analyze the results.
Okay, so did you guys encounter any problems while you all were doing this experiment?
We were debating on how much calcium to put in. And at first we had distilled water, but then we realized it was slightly acidic, so we used buffered saline instead.
So is your experiment ready to fly, or has it flown already?
It's gonna fly in December.
Okay, wish you guys the best of luck on your experiment.
Thank you. Thanks.
And what exactly is your experiment on?
My experiment is testing the effect of microgravity on ethyl fermentation rates.
And throughout your experiment, what problems did you encounter, and how exactly did you overcome those problems?
Actually, I was on the first mission, and in the first mission, it was a complete failure because there was leakage in the tubes, and the astronauts broke it at the wrong time. So the second time around, we fixed everything, had everything down, and we ran into no problems throughout the whole experiment.
Interesting. So, what is your next step? What are the next steps that you hope to take with this experiment or this project?
I'm gonna go wherever it takes me. Hopefully, I get to publish my results wherever I can.
Good luck to you, and I hope you the best of your experiment.
Thank you.
So what exactly is your experiment showing us today?
Our experiment is demonstrating that synthetic HBOCs, hemoglobin-based oxygen carriers, or red blood cells, can indeed function in space.
And throughout your experiment, what has been the biggest problem that you came across, and how exactly did you overcome that problem?
I think the biggest problem was the first time we sent it in, it didn't really get picked, and so they gave us some feedback. What we did is we just researched a little more, tried to make it better, got more information, sent it back in, and we got it in and we won. So.
All right. So what are the next steps that you hope to take with this experiment?
Well, we definitely want, if we could, we want to repeat this test and send it up to microgravity once again. But this actually helped us a lot, and we learned so much as a team.
Good luck in your experiment and your future endeavors.
Thank you.
So what exactly is your presentation on today?
So my presentation is on the effect of microgravity on the oxidation of iron exposed to a saltwater solution. Basically, rust in space. So if we find that iron rusted in space is weaker than iron rusted on Earth, then it can change how engineers design manned spacecraft because they will have oxygen and water inside of them.
Interesting. So is this experiment scheduled to fly? Has it flown already?
No, it has not flown yet. It's going to fly, I think, sometime in November.
That's pretty good. Well, I hope you the best of luck in your experiment. Hope you find out everything you need to find out. Good
Thank
job.
you.
Well, can you guys tell me the purpose of your experiment?
Our purpose of our experiment is to by conducting it in microgravity, we want to find out how to stop, grow, or even control mold differently here on Earth.
Okay, so how did you all go about completing this experiment?
Our procedures was first put the Gerber graduates for toddlers little biscuits 2 grams inside the nanorang test tube, volume 2, which will be separated by a clamp. Then pour the 2 milliliters of Aquafina distilled water into volume 1. And then finally, after 8+2 days after the arrival, the astronauts will slowly open the clamp, letting the distilled water contact the little Gerber graduates for toddlers little biscuits to start going on.
Okay, so did you guys encounter any problems while you were doing this experiment?
Yes. One of the problems was that we were going to use bread, but the NASA regulations didn't let us because when we would already be sending it up into space, it would already have grown mold and it would be into crumbs.
Okay, so changing your type of bread is how you all overcame your problem? Okay, so has this experiment flown or is it ready to fly?
It's not ready to fly. It's supposed to fly somewhere in December.
Okay, well I wish you guys the best of luck on your experiment.
Thank you.
So, explain your experiment to me. What's your hypothesis?
In my experiment, it's going to be the effect of tin whisker growth on a lead-free solder test bed.
All right. So, do you want to explain to me a little bit about what that means?
Basically, on a lead-free solder is 60% tin, and that is. It used to be 60% lead, but they've switched that out because Britain has a program that denies lead in solder. And the problem with this is it's a weaker material, and as it shoots up into space, the G-force kind of makes it crack, and a crystalline structure will appear out, creating its own circuit, causing a whole structural failure and short circuits to come.
Okay, so do you have a solution for this plan, or are you testing to see what it's all about?
Well, we're kind of testing to see the growth rate and see if it's different in microgravity, using that as an independent variable. The only solution they found right now is basically use lead solder because it's heavier and more dense.
Very good. And have you tested this in space?
Well, we did not get selected for the finalists of the SSEP, but my group personally won't take no for an answer, so we actually are still going to space. We're doing something called CubeSat, and it's a cube, a small cube that they cut in half and you put your experiment in, and they shoot it up into space for 5 to 6 months, and then it'll come down and you can retrieve all your data.
Very good, very good. Thank you today. That was a very good experiment. Thanks for coming by.
Okay, so can you guys tell me a little bit about what your experiment was on?
Well, we basically sent spider XX to space to see if they would change in physical appearance.
Well, for this experiment, we actually wanted to do crystals, but seeing as other students were actually planning on doing the same thing, we wanted to do something different. Well, just later on, I would look at my basement and seeing that it's invested with spiders. So I came up with the ideas of using spiders for our project.
So can you guys tell me the overall purpose of your experiment?
Our overall purpose was to see if microgravity could actually affect the development of a spider in space. And what we wanted to do is we wanted this to maybe help in finding out if this could also affect humans.
Okay, so you guys mentioned that you put them up in space before, so your experiment has flown?
Yeah.
Okay, and how did that go?
Well, the spiders did not come back alive as we wanted, but a very large spider was found.
it was actually dead, and it still had meat in it, which meant that it recently died.
Okay, well, it sounds great. I wish you guys the best of luck on your experiment.
Thank you.
Thank you.
And what is your presentation about?
The coagulation of erythrocytes. We're trying to find out if the erythrocytes will last longer in a microgravity environment other than on Earth.
And what exactly did this experiment show you?
Finding how long the erythrocytes will last in a microgravity environment will lead to certain experiments such as missions to Mars.
And what problems conducting this experiment did you come across?
Finding the blood, since it is human in origin, we did have a problem getting it all together and getting the equipment to test for the certain.
What is the next step you're planning on taking with this project?
Since we partnered with the American Red Cross to get all of the blood and everything, they decided to pull out since we weren't able to fly. So our secondary was to use propydium iodide instead of trying to find the technetium 99M, chromium 51, and indium 111 as a viability, cellular viability.
Pretty good. Well, I wish you the best of luck in your future endeavors.
Okay, so can you tell me the purpose of your experiment?
We wanted to test the rate of two metals out in space, iron and copper, and to see how that could be helpful in future space exploration.
So can you tell me the main things that you all did in order to complete your experiment?
What we did was that we prepared our materials and we thought of our proposal and objectives for our experiment. We looked at different types of metals that would apply to our experiment, and from that then we started on the proposal process and contacting such as teachers and other students to see if this was a project going forward.
Okay, so can you all tell me any problems that you may have encountered while doing this experiment?
Well, actually, whenever we got it back, we found out that only the iron they only activated the iron, so the copper was still actually not activated at all, and they were supposed to be activated and mix well with the water. So what we had to do is we had to set up another ground truth experiment, and we had to wait, and then we analyzed those both and saw them side to side and compared them.
Can you guys tell me what conclusion you all reached?
But from the information that we did receive, we found out that the iron actually oxidized faster in space than it did on Earth, which contradicted our hypothesis.
Okay, so you guys mentioned every time you sent it up, so how many times has it flown?
It's flown once, and it's going to fly again this fall.
Okay, that sounds great. I wish you guys the best of luck with your experiment.
So what exactly is your presentation talking about?
We're testing to see the effect of microgravity on the rate of cell division.
Okay.
Yeah.
All right. So what exactly do you hope to achieve with this experiment?
Well, cell division is like an important part of life. Like all the cells undergo cell division to reproduce, to grow, and to heal itself. So that's important for life in general. Well, we're hoping to see whether or not microgravity will affect cell division negatively or positively, and then that will help us determine the healing process and how it can go on in microgravity.
What experiments did you actually conduct?
On the ISS, there's no incubator, and that's usually where the cells grow. So we tested room temperature and incubator growth of the cells, and we found that incubator was more favorable, and then room temperature was not as favorable. But if you start out with a little more cells, the growth is better.
Okay. And what problems did you have while testing that experiment?
It was really difficult to find cells that we could use because we're not certified to purchase cells or experiment on cells. So we had to find someone, a doctor at City of Hope Cancer Research Center, and she donated cells for us to use for the experiment.
Interesting. So when is this scheduled to fly?
September.
All right, September. Well, I hope you the best of luck, and hopefully your experiment would actually work in space. And what exactly is your experiment on?
My experiments on silver crystals, they are a metal organic framework, which means that they can absorb and store gases, let's say in a space shuttle or a vehicle, and they could create an innovative gas tank that could hold more fuel than ever before.
All right, interesting. So throughout this project, what has been the biggest problem that you had to actually overcome?
We faced a problem where we didn't know which crystals to use. We had many different types that we could use, but we discovered that these had the best applications.
All right, interesting. So has this project flown yet? Is it scheduled to fly?
This project will be flown in the fall. It's Mission 4.
Mission 4. Well, hopefully it performs exactly how you thought it would in space. Good luck.
All right, thank you.